Hello and welcome to this podcast for cell biology. This is our last podcast of the semester. And in this podcast, we're going to continue our discussion of the cell cycle and the regulation of the cell cycle. But now we want to talk about what happens when that regulation goes wrong. How those cells, which normally respond to certain signals, stop responding to those signals and begin to grow aggressively and ultimately form a tumor and then metastasize into a cancer. But I want to begin by showing this video. As we watch this video, the very first part of it is going to show healthy cells taken from breast tissue. You can see them starting to grow a little, some divisions here, they're getting a little larger, the cells. Here they circled this one. You can see, if we hit pause, how the cells are distinct. But you'll also notice another thing, that as it's growing, this fairly well-ordered spherical structure. Not a perfect sphere, but it's not irregular. Over here you can see a very similar one as well. When we move to looking at a cancer cell, you'll see how much different that is. Now what we're going to look at are cells taken from a breast cancer patient. And as we watch this, you're going to notice a couple things here. I'm going to pause it here. You're going to see that the structures forming are going to be very irregular. And we'll let this go and it's going to become even more irregular. And you'll also notice how much faster they're dividing than the previous ones. And that's one of the hallmarks of cancer, this very rapid cellular growth. So let's watch this. And remember, this is in the same time frame as the healthy cells. And you'll see they form these large clumps of cells, as you see here. And I'm just going to show this one more time here, the exact same video, but it's going to repeat it here, so let's just watch it. And so this large clump of cells, when someone is diagnosed with a breast tumor initially, they will often feel a lump. And you can see how this lump could form from these cells growing rapidly and very irregularly. So this could easily form into what would be classified as a lump in someone's breast. Okay, now let's go back to the whiteboard. Okay, so let's begin our discussion of cancer. And we'll begin with a simple definition. We can initially think of it as a disease caused by uncontrolled growth. But I want to add something to this. It's not just cells growing out of control. For it to be a cancer, those cells need to metastasize. Cells need to metastasize. Now I'll talk about what that means in a little bit. But until then, you should know metastasizing cells means they just go from one site in the body to another site in the body. It's a really important disease for us to talk about. Probably everybody in this classroom has either directly or indirectly been impacted by this disease. And so just some quick stats about cancer. It's the second leading cause of death in the United States. Heart disease is the first leading cause of death. We know that there are about 200 different types of cancer. So unlike most diseases that we may talk about, cancer cannot be lumped in as a single disease. And this is a rather striking number. We know that about 40% of Americans will develop cancer at some point in their lifetime. You can inherit a predisposition for cancer, but most cancers, there is no hereditary link to them. So they don't necessarily have to run in a family. They can, but they don't necessarily have to. Linked to this, we know that 2 points I want to make about this is, the first is that for a cell to ultimately become cancer, it requires approximately 10 different genes to be mutated. So if you're not inheriting all those 10, and you wouldn't, most people, if they inherit any, they inherit one, maybe two. But inheriting just one sets the stage for the potential to form cancer later in life. So where do these other mutations come from? The other 9 or maybe all 10 of them. They come from the environment, largely, not exclusively. Or the other part comes from errors in DNA replication. But cancer has a strong environmental component. Certain environmental factors will increase getting certain kinds of mutations. I'm going to list something up here that I mentioned for this inheritability of a predisposition factor. Some reports I've read it's only 5 to 10% of people with cancer have a hereditary link. Again, the environment plays a huge role. And because of that, prevention also plays a key role in our fight against cancer. And the last thing I want to say here is that of these 10 mutations that have to occur, the first mutation almost always affects cell cycle regulation. So I want to continue my discussion of these common characteristics of cancer, but I'd like to begin by drawing it out. And what I'm drawing here are skin cells. They could be other cells, but let's go with skin. And of course, all of these skin cells have a nucleus in them. And initially, there's nothing wrong. But maybe there's some UV damage that comes along here. And maybe it damages the DNA in this nucleus. So I'm just going to color that in. And as I said, this first mutation may block some part of the cell cycle. So maybe it mutates a gene that's necessary for this G1 cell cycle checkpoint. So maybe this mutation that stops this cell cycle checkpoint at G1 right here no longer allows this cell to check for mutations. And if it's not checking for mutations, it's able to go through this cell cycle and copy its DNA, even though it's mutated. And now what's going to happen is that several other mutations will now occur in this cell and the progeny of this cell, the daughter cells of this cell. And at some point, it begins to transform and no longer will look like a skin cell. So now what I'd like to draw is this same layer of epithelial cells, but after many generations of division. But now, these cells have lost their normal appearance and they began to take over a little bit. They're growing faster now because they no longer have this stop point. So they're growing faster than their neighboring cells. And again, I'm trying my best to draw them so they're looking a little different. But a key point here, a couple key points, they're growing faster, so they're outcompeting these cells for space as well as nutrients because they're dividing faster. We would call this a primary tumor. And at this point, we would call this primary tumor benign. Benign because it's still in the same tissue where it started. However, what's going to happen as this continues to grow faster and out of control, and it's also important to take note here that during this time, every time they divide, because they're blowing past this checkpoint, they're accumulating mutations. These mutations allow them to continue to grow faster, but also to escape these tissues and to escape things like the immune system. So what's going to happen next is that these cells of this primary tumor are going to invade the tissue below it. And I'm just going to draw these cells slightly different. And these are healthy cells. They're just in a neighboring tissue. But soon, these cells will begin to penetrate these tissues. And because they're now growing out of control, they will begin to take over this tissue. So now at this point, these tumors, these benign tumors, they're going to start to affect these tissues that they're invading. And that can be problematic because now these particular tissues are being taken over by these rogue cells dividing out of control. So these tissues will be directly impacted. But the real problem comes next, after they've accumulated even more mutations as they spread beyond this primary site. And what they're going to do is something that is kind of clever, but is also quite scary because of the result of it. There may be a blood vessel along here that's doing its normal job. But what these tumor cells will begin to do is they will begin, these as well, begin to secrete chemicals that are going to interact with this blood vessel. And as they interact with this blood vessel, they will cause this blood vessel to grow in this direction of the tumor, forming different offshoots. So that now a couple things are going to happen here that can have very devastating effects on this person. These cells here are now recruiting their own nutrients so that they can continue to divide. Usually, growth of our healthy cells are limited by the space and the amount of nutrients they have, but they are now pumping nutrients right to the cells. This benefits these cells, these cancer cells, and their ability to grow, a benefit at our cost. Another thing that this allows the cancer cells to do, it allows them to escape this primary site, and they can enter into this circulatory system. And as they enter into the circulatory system, these cancer cells can enter into other tissues of the body. We're getting a little tight on space here, but let's just draw a tissue. So if this started off in the skin, let's say now we're in the liver. And now these cells, because the liver itself is highly vascularized, it provides a path for these cancer cells to enter into the liver. So once in the liver, they will escape the circulatory system confines here, and now they will start growing in the liver, outcompeting the healthy cells of the liver, and now they will eventually push these healthy cells out of the way, and now the liver's function is going to be impacted. These cancer cells can continue to invade other parts of the body as well. At this point, once it spreads to other parts of the body, we say it is no longer benign, but we say it is malignant. Cells that have left a primary tumor site and have entered a secondary tumor site, we say that tumor is now malignant. So now I want to go back here to this initial cell that receives that first mutation and talk about the proteins that are involved that ultimately lead to this. So let's come back to our cell here before it became mutated. Now this cell, like most of our cells, is only going to divide when it receives a signal. This signal from another part of our body. Remember, we talked about this social control, that other tissues in our body, some kind of gland, usually a pituitary gland, adrenal gland, various glands, secrete some sort of signal that then binds to this receptor and tells this cell to divide. And that's what it does. It'll form two new cells, but only when it's supposed to. So I want to talk about what it's doing normally first, like it's supposed to do. So I'm going to make this cell a little bit bigger, and I'm going to introduce some new proteins for you. So here's our large cell here. And remember, I said it has this receptor on the outside that will respond to some sort of signal. But for right now, there's no signal. So the cell should not divide. The way it's going to divide eventually is when this protein called E2F. I'm going to make a little key up here. The red one here is going to be E2F. So when E2F enters the nucleus, what it's going to do, it's going to bind to a gene. And when it binds to this gene, it's going to serve as a transcription factor to turn on genes necessary for S phase. But only when it has this signal that binds to it will E2F enter the nucleus and turn on these genes necessary to go from G1 to S phase. So for now, let's take E2F out of the nucleus. So this gene right now is not turned on. So we are not making proteins necessary for S phase. And the way the cells keep E2F out of the nucleus is by having it bind to, you guessed it, another protein. And this other protein is a protein called RB. It stands for retinoblastoma. As long as RB is bound to E2F, E2F cannot enter the nucleus. Okay, now it's time to signal the cell to divide. And so what's going to happen is this signal is going to bind here. And when it binds here, it's going to cause a sequence of events. It's going to take a CDK. Now it's a different CDK than the one we talked about, but CDKs always are regulated by cyclins. So again, it's not the same one that we saw as the G2 checkpoint. This is one that's involved in the G1 checkpoint. Initially, this is phosphorylated at both sites, so it's inhibited. But once the growth factor here binds, it causes a set of events that will remove that phosphate group. Once this phosphate is removed, I'm just going to erase it. Now it is active. This being active, it will now act upon RB. So let me draw the arrow over to RB. Remember, CDK is a kinase, so it's going to add a phosphate group to RB. When it adds a phosphate group to RB, a conformational change occurs, releasing RB from E2F. So now E2F has been released, and now it can enter into the nucleus. Once in the nucleus, it will bind to this promoter, turning this gene on, but only when this is bound. So this is under healthy, normal circumstances. We only turn genes on for the S phase when this growth factor has bound to it. But on our last whiteboard, I showed you this cell and said it gained a mutation that forced it into S phase, even though it wasn't supposed to go there. So to talk about that, I need to introduce one more protein. It'll be the last one of the semester, I think. And it's P53. P53 has a very interesting job. It detects mutations. And when it detects mutations, it either fixes the mutation or sets up a pathway to destroy the cell. So even if this signal is bound here, and the cell should transcribe genes to go into S phase, P53, if it detects a mutation here, so another part of the genome, if it detects a mutation, P53 will find that mutation, and then in an effort to fix it, it will keep this cyclin CDK complex inactive. There's more to it than that, but that's what we need to know for now. P53 detects mutations, and even if it's signaling the cell to divide, P53 keeps cyclin CDK inactive. And as long as cyclin CDK is inactive, it won't be able to phosphorylate RB. If it can't phosphorylate RB, E2F will remain in the cytoplasm and you won't move past S phase. So that's the job of P53. Finds a mutation, stops the cell cycle right here at G1, stops the cell cycle by preventing this complex from doing its job. Then it fixes the DNA. After the DNA is fixed, it will remove its inhibition on here. We will remove the phosphate group, and now we can phosphorylate RB, release E2F into the nucleus to turn these genes on. Now imagine a scenario where P53 is mutated, so it's not in the cell anymore. Yet we have a mutation here. Normally P53 would stop this, but since P53 isn't around to do its job, to fix this mutation, when that growth factor binds to this receptor, it will cause a signal pathway to activate cyclin CDK by removing that phosphate group, even though there's a mutation that should have been fixed. Now, cyclin CDK is active. It will add a phosphate group to RB, releasing E2F, and now with E2F released, it moves in here, binds here, and turns these genes on, and now we move to S phase, and we copy the DNA, including this mutation. We will go through S phase, G2, and mitosis, creating 2 new cells, each with this mutation. And now this mutation will be set in that cell, and every time it divides, it will carry on that mutation. And since P53 is no longer in these cells, you'll never stop it for any additional mutations. And now this cell can begin to grow without regulation. And it can form that primary tumor. It can then gain enough mutations to invade neighboring tissues, to attract blood vessels, and then to metastasize to other organs in the body. P53 turns out to be an incredibly important protein in regulating the cell cycle. All right, that ends our podcast for cancer biology and the cell cycle. And sadly, it's the last of all of our podcasts for this semester. I hope you've enjoyed it, and I hope you've learned a lot. If you have any questions, of course, about this material, but about anything as you move forward at CMU, please make sure you don't hesitate to stop by and chat with me. Good luck. Bye.
Until we meet again, happy trails to you. Keep smiling until then. Happy trails.